unravel the causes of birth defects. I want to make sure that you understand that this, although I have many slides, I really want this to be interactive. There's no intent on my part to get through all the slides. If you have questions and you want to bring those questions and present those questions throughout, please do so. Because I want this to be something that is valuable to you. And if I just stand and lecture and you would prefer to be asking questions, then let's make sure that we do that, okay? So I'm going to start with a story. And the story begins in 1928 in a little stone house in close to Ottawa, Ontario. And it was the house where Clifford and Maggie Hammond lived. And they were about to have their fourth child. Maggie was pregnant, and she told Clifford to go and get the doctor, that it was time. And Clifford went to get Dr. Jones, and he came to the hospital, came to the home to deliver this baby girl. And he went into the house and proceeded with the delivery. He came out quite a bit later after Clifford had expected, and he was shaking his head, and he said to Clifford, you know, your wife is fine, but I'm really not sure that your baby girl will survive. She has been born with a problem, a birth defect. She has her intestines are outside her abdominal wall, and we have a choice right now. I can try to do some surgery that I've never done before. I just read about it, but I've never done this kind of surgery. We could take her to the hospital, but quite frankly, you know, it's by the time we get there, we have to take horse and buggy. By the time we get to a local hospital, I'm not sure that she would actually survive at all. Or I can just do nothing and she will die. And so Clifford said, well, let's go ahead and why don't you try to do the surgery, and we'll see what happens. And so he did the surgery. The baby girl survived, but she was in critical condition for several weeks thereafter. It took forever to get her to feed properly, and so there was a lot of concern that she really wouldn't make it. Well, anyway, she made it, and she became a teacher. She met her husband, and they got married at the same home in which she was born, and they were married in 1952. And then they went on to have pregnancy themselves. They had 7 pregnancies, and with those 7 pregnancies, their first baby was a full-term baby, but was born and was really dead upon arrival, so it was a stillbirth. Without any explanation, they had 2 pregnancies with miscarriages, but they eventually had 4 children that were live born, and I was the second of the 4. And so it led me to understand that the miracle of healthy babies and healthy births is something that we cannot take for granted. And knowing that, and then very much wanting to study medicine and study especially children's health, I went into the area of birth defects. So, and I tell you that story to let you know that almost every one of us will be affected by someone. Either we ourselves will have been affected by a birth defect, we'll have someone close in our family or close to our friends that we know has a birth defect. And I'm defining birth defects in this context as a structural malformation, something that happens within the time between conception and the first 10 weeks of following conception, such that the parts of us, if you will, the hard parts that come together in each organ system don't come together in the right way. So the building blocks somehow don't come together in the right way, resulting in problems in multiple different areas. For example, a cleft lip or a cleft palate is a problem with the formation of the mouth. Babies born with spina bifida, it's a problem with the formation of the neural cord. The most prevalent kind of defect is a problem with the heart, so congenital heart defects. But every single organ system that you can think of, the building blocks of what makes us human, can come together in a perfect way so that baby's born and you walk into the delivery room and there's a healthy baby, or you come into the delivery room and there's something wrong. And I do my clinical work at our university hospital here where we do deliveries, and of course it's really not, it's every week that we will have a baby that's born with a birth defect. So globally, 8 million babies a year are born with a major birth defect, and 3.3 million will die before their fifth birthday, and as I've said, heart defects are the leading cause. So what are the causes of birth defects? We know that things that do not cause birth defects, for example, if you look in the history of medicine and texts from the history of medicine, you will hear such stories as, you know, that we once believed that women who came across a rabbit when they were, you know, 6 months pregnant or so, and there was some interaction with the rabbit, then if that, then if they have a baby that has a cleft lip, then that's where your lip came from, right? I was just in Rwanda and teaching a course for the American Academy of Pediatrics in Rwanda. I just got back in September, and in that culture, I was able to see babies that did have very severe birth defects, but it was still thought in that culture, especially in the very rural areas, that these babies were born with a birth defect because of something that mom did wrong. It was kind of a damning, a divine damning, if you will. And of course, we know that that's not true. But we think that it's both environment and hereditary factors. There are some birth defects where we know that there is a single gene disorder, but for the majority of them, the majority of birth defects occur. We really think it's multifactorial, both the environment and genetics working together. When we can identify causes, then we can help to prevent birth defects from occurring, but we have to identify the cause first. So some of the challenges of finding causes of birth defects, the temporal relationship, right? You can imagine that there is an inherent bias or an inherent challenge to find the cause of birth defects. You know, we have to rely on, those of us that study human health have to very much rely on scientists who study animal health, especially when it comes to birth defects. We're not going to interrupt a human pregnancy to study birth defect, but we can interrupt an animal study to better identify the mechanisms that might cause birth defects. We're not just dealing with one patient, we're dealing with two patients. There's both maternal factors and potential fetal factors. There are limitations with our colleagues that are studying animal models. We can't always just presume that you can do a one-to-one relationship. And we know that for about 70, or we hypothesize for about 70% of all birth defects that it's due to a complex etiology. This is Clark Fraser. Clark Fraser studied at one of my alma maters, which is McGill University. He was a famous teratogeneticist. And when I was teaching courses there in the mid-1980s and talking about craniofacial malformations, I would always invite Dr. Fraser to come to the class, and we would talk about the multifactorial etiology of orofacial clefts. And we would say, well, it's both environmental factors and genetic factors, and we would say, so what are some of those genetic factors? And it was pretty much a black box. It really was. You know, we could say, that was about it. We could say there's a familial aggregation, so that within the same family, there seems to be a preponderance of individuals with cleft lip and palate more than we might expect in the general population. But what those particular genetic factors are, we're not sure. There have been some successes when we come to look at birth defects. Those of you that are in the room that are my age or older will have had perhaps their mother having taken something like thalidomide, especially if you're from Europe. You know that it was used for nausea and vomiting. Thalidomide was a drug that was used to prevent nausea and vomiting in pregnancy in European countries and a derivative of that was used in Canada as well. Before we were able to see, it was really astute clinicians that together we were able to see that moms who were, women who were taking thalidomide during pregnancy were having babies that had no arms and frequently no legs as well. And so you may have seen pictures of that. So that was I say that was a success in that we recognized it. Many would say too late, but at least it was recognized. Folic acid is a B vitamin. We know that if women will take folic acid prior to their pregnancy and during their pregnancy, that they will be less likely to have a baby with spina bifida, which is a herniation of the spinal cord. And then rubella, we know there's congenital rubella syndrome, and that's really one of the reasons this individuals are now immunized against rubella. Again, in Rwanda, where I was, they have just started their rubella immunization, and their hope is that they will have a decrease in congenital rubella syndrome. Accutane is just an example of a medication where Accutane is used for treating adolescent acne, and we know that it can cause terrible embryopathy, terrible birth defects. And now if you have a son or daughter or a loved one who's on Accutane, they, at least in the United States, I expect in Europe as well, that you have to be on in a registry. These teenagers that take it have to take pregnancy tests every month to ensure that they are not taking Accutane and at risk of becoming pregnant at the same time. Some of the challenges we still have, we know that one of the strong risk factors for birth defects is maternal diabetes, type 1, what we used to call childhood onset diabetes, and type 2, and with the growing epidemic of obesity in our countries, we know that there are more and more individuals with type 2 diabetes, so that is a real challenge. Obesity, even without diabetes, is a risk factor for birth defects. It must be something about that glucose metabolism that causes something to go wrong when the embryo is initially developing. Animal models have very strong evidence to show that as well. We continuously have new medications on the market. You know, how are we going to have another thalidomide scare? We need to be watching this. And then you have the woman who comes up almost, and I have to always remember this because when I'm giving a talk to this, especially to the general public, there will always be a woman in the audience who will come up and say to me, I have a baby with a congenital heart defect, or I had a baby who passed away. I didn't smoke, I didn't drink, I ate properly, I exercised, my weight was appropriate, I did everything right. Why did I have a birth defect? And that's what we don't know, right? So it's really important that we remember that and that we don't blame the mom because maybe we think, oh well, maybe you did something wrong. We don't want to get back to that era at all. So this is just a schema to depict that we think that there are genetic factors, environmental factors, there are innate immunity factors, hormones, and then epigenetics. And epigenetics we'll talk about in a bit. Clyde, may I also ask that you give me 5 minutes before the end when you want me to finish, so that, like just a time, so that in the United States, because of the concerns about birth defects, there was a congressional statement that was made in 1996 that said, we really need to understand and identify causes of birth defects. What are we going to do as a nation? I must say, and I want to make it really clear, that a lot of the very good work that has been done identifying causes of birth defects has been done in Europe. A lot of the work was done in Hungary, and it continues to be done in Hungary. One of the randomized control trials that helped us to identify the real importance of folic acid was done, it started in Britain and was done in multiple countries, but there's been a lot of really good work done in Europe. In the US, our way to address it was to identify centers that could be involved in etiologic research regarding birth defects, and so these are the centers that are up on the map here. Arkansas, we were involved very early on, and we were very pleased to be involved. Again, back to congenital heart defects accounting for about 30% of all birth defects. 1 in 4 babies born with a congenital heart defect will die. Complex etiology. And we think that multivitamins not only helps to protect against neural tube defects, but also helps to protect against congenital heart defects. We have a large program taking care of babies with congenital heart defects here at our hospital, the Arkansas Children's Hospital. And so this is just a picture of some of them. Because we are a fairly rural state, often the babies are born out in the state in a smaller hospital, and we have to use our helicopter and our transport system to bring these babies to us. Ideally, we would like it if the babies are prenatally diagnosed. Only about 50% of them are prenatally diagnosed because it requires a special echocardiogram of the fetal heart. And so we really want these babies to be born in Little Rock if possible, so that we can transport them very quickly from the hospital where I work over to the Children's Hospital where I do my research. Yes?
Are the centers arranged geographically by capability or both?
The centers were chosen by, the prerequisite was that they had to have a population-based birth defect registry, which is like a public health registry that goes out into the defined population, and is able to identify babies with birth defects. So it was competitively awarded, but the prerequisite was that you had to be covering a population that had at least 30,000 births per year. And so at the time, there were not a lot of states that had that registry. It's fairly expensive, and it had to be active in that we had to have staff that went out to each hospital to identify and collect medical records. So that was how it was done.
Just epidemiologically, how is the prevalence of birth defects in Europe and in the US?
So in Europe and the US, there are some clear differences that are sometimes related to etiologic differences. For example, we have more obese, right? More diabetes, right? But then there are some differences where you have a lower population. In France would be a good example. I don't know if there's anyone here from France or not. But in France, and there may be other countries in Europe too that you would say, well, that makes sense, that's what we do as well, that there's more likely to be prenatal diagnosis followed by termination. So, for example, Turner syndrome, which is a problem with one of the chromosomes, or gastroschisis even, where the baby's born with the intestines outside the abdominal wall. It would be much more likely in France to terminate that pregnancy prior to the birth of that pregnancy, where in Arkansas, for example, in some of the states that are in the southern, especially the southern Bible Belt states, we would be much less likely to terminate those pregnancies. So you have some differences, cultural differences there. This is a story of Benjamin. He was born on 2008 to a couple who, at the time, the dad was a pediatric resident and mom was a medical student. And they went in, they had a prenatal diagnosis. They knew that Benjamin had hypoplastic left heart syndrome. Mom stopped going to medical school when the baby was born, because they're told that having a baby with hypoplastic left heart syndrome that can take on the family is amazing. And then the cost, of course, is very, very high. But you couldn't really, one of them had to stop doing what they were doing, right? You couldn't, it would be almost impossible for both mom and dad to continue in a medical career. Now, they've since picked that up again. And I'll tell you more about Benjamin later. So this was the National Birth Defect Prevention Study that we did in all of the states that were funded. It went on from 1997 to 2012. There were 8 sites. We did over 44,000 interviews. So we, as a group, collectively identified babies with major structural malformations. We contacted the mom, and then we called the mom and did an interview with them to see what kind of exposures they had during pregnancy. At the end of the interview, we asked Mom if we could send her out a kit so that we could collect from her samples from the cheeks that were cheek cell samples that she would send back to our laboratory so that we could then start looking at some of the DNA factors or the genetic factors that were related. And so here would be a typical question that we might ask during the interview with the mom. Did anyone in your household smoke cigarettes in your home from 3 months before you became pregnant to the end of your pregnancy? So we would get information like that. Again, it's a kind of, some people would argue, well, that's a kind of crude measure. Is she going to tell us the truth or not? There would be better measures, right? You could measure cotinine or a biomarker in the blood to see whether or not that was the case. But the resources were only available at the time to do it. So there are limitations. In Arkansas, this would be the percent of women smoking during the first trimester. These are the other sites. In Arkansas, I'm hesitant to tell you, but have to, that about 28 to 30% of our women smoke during the first trimester. That's pretty high. I don't know what it would be like in other countries, but it is certainly higher in Arkansas than in other states. And we know that that's not good for the development.
What about the second-hand smoke?
Secondhand smoke, so then we asked about whether they were exposed to secondhand smoke as well, and if you have that, so both the active smoking and the passive smoking, about 42% of women. So it's quite high. And so then we looked at these data and were able to say that yes, there was an association between smoking and the risk of cleft lip and cleft palate, and that there was also a risk of maternal smoking and congenital heart defects as well. And then we went on and found more information about obesity, as we talked about. And then we did talk about spina bifida and folic acid, but we also found an effect, the protective effect of folic acid against the occurrence of congenital heart defects, which was very important. So now to talk a little bit about genetics, yes?
This may not exactly be the time, but recently in the I'm not in medicine at all,
Okay.
but recently in the popular literature, it kind of came out that multivitamins were a waste of money.
Right.
So what do you have to say about that?
So one of the things that happened, the United States Public Health Service in 1992 said there's sufficient evidence to say that folic acid is a good thing, that we should be taking, that women of childbearing age who are capable of becoming pregnant should take folic acid supplement. And I frequently speak to trainees somewhere along their, medical student, a resident, a fellow. And if I'm talking to, if I'm giving a lecture and I say to the medical students, so how many of you took your folic acid this morning, among the women? It's usually about a third took a folic acid supplement within the last 48 hours, right?
I just thought it was kind of a poor recommendation, because for a nickel I take my multivitamin. I'm sorry.
Right.
And they're saying it's a waste of money. It just
Ring
true somehow.
It doesn't ring true, and it doesn't, and there are many other things that multivitamins can do for us, right? Yes.
You study multivitamins almost kill things.
Okay.
There apparently are some individuals on take multivitamins that they don't do one. I assume you guys look into those kind of factors too.
So we will ask women why they don't take it, because only about a third take it, folic acid, and they will tell us that weight loss, vomiting, that they don't feel as well, that there are other factors, and that needs to be really taken into account. Why is it that some people can take multivitamins and do well and others not? Should they be taking a single folic acid supplement? If you have had a baby with a neural tube defect, you're 10 times more likely to have a baby with a subsequent pregnancy that's also affected by a neural tube defect. In 1998, the Food and Drug Administration in the United States fortified foods, rice, grain, and cereal products with folic acid. So if you look at your nutritional supplement or the label on your foods before 1998, you won't see folic acid, or you'll be less likely to see folic acid. But if you look in, say, cereals, after 1998, you're going to see that there's folic acid put in the food supplies. If you look at studies, national studies that have measured folic acid in blood before 1998, that level of folic acid is less than if you look at similar studies where we look at blood levels of folic acid in the population. It's higher because of the fortification. So that then, some people said, well, do they still need to take supplements? Because some women are saying that the supplements are causing them harm. And I would still say that if my daughter was pregnant, I would want, or before she became pregnant, I would want her to take folic acid. I think the evidence is potentially, there's some controversy about it, but I would still go ahead and take the folic acid, unless for some reason, at an individual level, not a population recommendation, but at an individual level, there was really compelling evidence that folic acid was not working for that person. And we want to get to personalized medicine, and we can only get to personalized medicine by some of the studies that are related to genetics. So you may have a genetic susceptibility to not being able to process multivitamins well.
Destroyed my liver.
Right.
And I am part of the Veterans Administration is doing I think there's almost 2 million of us now, where we all volunteer to give our DNA and they're gonna follow us for years.
And that's wonderful, because then we may find out, why is it that, and can we be more precise in the recommendations that we give people? Because as I said, right now it's all women of childbearing age who are capable of becoming pregnant. And that seems to me kind of a crazy recommendation. First of all, if I am not sexually active, then you're kind of saying, well, I need to take it anyway, because that is a recommendation. The comment is, I might be of a different sexual orientation. I mean, there's all kinds of reasons why that blanket statement is not appropriate. This is Watson and Crick, and of course, Watson and Crick, with the help of many before them, including, I've forgotten her name right now, I remembered it last night, but the famous woman radiologist who was able to show the physical structure of DNA. But Watson and Crick are the ones who are really shown to identify at least structure of the double helix DNA. And after that was done, there's multiple other scientists, clever people, who worked hard to be able to sequence the first human genome. And it was done by the Human Genome Project that was funded heavily by NIH, but other countries as well participated. And in 2002, 2003, we had the first sequencing of the human genome that took about 10 years to complete. We've gone on to do studies where we can say, for example, here we're just looking at, and this slide is a little dated, but if you look at humans, the number of base pairs, right, the number of nucleotide base pairs is about 3.2 billion. The number of genes estimated at 25,000, really it's about 21,000 now, I think would be more likely to say. The number of chromosomes, 46. So that's what we're looking at when we are thinking about what do we need to do within the human genome. And of course you can look comparatively across other different species. So what is a gene mutation? Because we think that potentially it is gene mutations that cause diversity and potentially cause some of these things that may go wrong in the infant as they're being developed. And so there are changes in the DNA code, right? And there will be one of the nucleotide bases that will be missing, added, or exchanged. And how do they affect a population? Well, they are a source of our variation, and some may be better able to survive than others. So a single nucleotide polymorphism, there are different kinds of mutations. The single nucleotide polymorphism has had a lot of attention because it's one of the ways that technology has advanced to let us look at genetic variation. So if I have a substitution of one of my nucleotides for, in this case you'll see a C to G, this is the one that we're looking at here, just a C to G, can you see that? Yes. And this is, and then a T to A substitution or a SNP there, that's a single nucleotide. They're the most common in the human genome, and we think there are about 7 million of these. Now, some of them have absolutely no impact on our proteins that are then made following that, and some will have an impact, and some may even have an impact on diseases. So I'm gonna skip through that. The HapMap project, following the Human Genome Project, again, was a major international collaborative endeavor where the major objective was to identify genetic variations across multiple populations. There were 4 distinct populations, but across multiple populations, including Europeans and Asians, Africans, and to be able to see how these variations and where these variations occurred in the genome. That tool then, that ability to identify variations, was then used to say, do these variations occur more likely in patients with, for example, a birth defect? Or do these variations occur more likely in patients who have cancer? And so that, with the tools of the Human Genome Project, the International HapMap Project, we were able to go into our lab at Children's Hospital Research Institute and begin to use some of those tools and some of those approaches to address more carefully the genetics of birth defects. I'm just showing here, you know, you think that it's rocket science, but it isn't. Although this machine here costs about $400,000 or more, it sits on the desktop, it takes about this much space, and it allows us to look at human genetic variation. There are a couple of vendors that have a monopoly, I would say, on this. One of them is Illumina, and as you might imagine, the technology keeps advancing at a very rapid pace, so that you purchase this for $450,000, and within 3 years or less, there's the next better model that can do even more. And so if you're in the game of science, which requires you to be competitive and to be able to do work that is really cutting edge, you are then faced with having to replace the work that you have where you're looking at maybe 6 SNPs, single nucleotide polymorphisms, to what the next Illumina is able to let you do. So the next one is 1,536 SNPs. We did that, and then the next one is where we are now, it's 5 million SNPs. So now we're looking at 5 million pieces of data. So since 2000 to 2014, we've gone to being funded to look at 6 SNPs in each of our children, to now looking at 5 million plus 250,000. So it's called the 5 million chip, but they've added 250,000. So 5 million single nucleotide polymorphisms in each child. So you can imagine when you start to hear people talk about big data, that's what we're talking about. You know, we're talking about an amazing exponential growth that is driven by technology. And then the question is, what on earth do we do with all those data? You know, how are we going to, that's why if you start hearing about bioinformatics and the need for statistical geneticists, it is because of the real increase in data. If I had a child that I could influence, and I have 3 teenagers, and believe me, I cannot influence them, but if I had a child who I could influence, I would be putting them into informatics. I'd say, when you go to undergrad, you can take music if you want. I think it would be great. We were just talking about the fact we don't have any musicians in our family, so it would be great if they could. But in our family, if they could take music plus informatics, you know, that would be wonderful, because we are in the day and age of big data, and our young people are learning how to use big data, but there are not enough young people in the field yet, because you get something like my son where I say, this is such a wonderful field, why don't you go into it? And he says, Mom, the last thing I want to do 8 hours for, you know, every day of my life is to sit in front of a computer screen and look at data. That is not what I want to do. But yet, I would still think of I'm still trying to think of novel ways to entice young people into this field, because it's so needed. there are multiple colleges, liberal arts colleges or universities that in the United States that have, even at the undergrad level, have informatics programs. So they may have bioinformatics as a department, or if they don't have that at university, they certainly just to get into it would be another way to get into informatics if there's not a formal program would be through computer science. Computer science would be, as an undergrad, I think would be probably one of the best ways to go if there was no formal bioinformatics program. And I'm not talking about that being their major. I'm talking, I've seen young people who seem to be more aware than others who will say, well, I'm really passionate about history. My goal is to work in maybe a library or to work in a museum, but I know that I'm going to need, I know there's going to be big data there too, and I know that I need to be very facile in terms of being able to work with big data, and that may come through computer science as well. But the informatics programs I see popping up all over the place. I don't know what people are experiencing in Europe, but association studies, if they were probably, we're still having in our lab where we do the 5 million, and I'm not sure if 23andMe does 5 million.
One.
Okay, so for 5 million, it's 240 now. The whole exome sequencing can be less, like about $99, which is a bit odd, but it's just a new platform that's come out since January that we're just purchasing. And whole genome sequencing is still about $3,000 to $4,000. And that does not include the analysis, right? So it just gives you the data from the lab, but without including the bioinformatician's time, the statistical geneticist's time, the genetic epidemiologist's time, the clinician's time. So we hear a lot when we go to genetic conferences that the hope is that, well, I don't know if it's a hope, but you certainly hear that the practice may come in the not too distant future where every baby that is born in a resource-rich country, like the countries that most of us are from, will be sequencing the baby. So the baby's DNA will be sequenced at the time of their newborn hospitalization. I think we're a ways from that, but I cannot, with the amazing advancement over just the 10 or 13 years that I've been in this field, I wouldn't be surprised that will happen within before I die. I mean, if it's going to happen, probably before I retire. And then we have epigenomic studies, which are studies where we're not looking at changes in the sequencing of the DNA, but we're really looking at changes in methylation, or things that happen that don't alter the sequence, but alter the expression of the gene. So if you think about the sequence as the letters in a story, the epigenomics you can think of as the punctuation in the sentence, or the paragraphs. And so the way that we would go about our studies, for example, is that we would get, for something like birth defects, because of the challenges, we have to identify patients that have the problem, get their DNA, get non-patients, infants that are healthy, and then compare the differences of their distribution of the variations in each of the group. And I'm going to skip through this.
Don't skip that.
We'll have to.
That's right up our alley.
When we think about candidate genes, this is the biological pathways. So these are all the biological pathways, if you will. And when we used to do just candidate gene studies, we'd say, you're just at that one spot. Now we do genome-wide association studies so that we can be agnostic about it. Sorry, but I want to get to, we've talked about this.
You might want to go into the question and answer.
Yes. One other thing that we haven't talked about are the jumping genes. And there are parts of our genome that can jump, and that's one of the new areas that we're looking at, and that is transposable elements. But this was discovered by Barbara McClintock in work that she was doing in corn, but she was able to identify that you can have a particular area of the gene that when it is replicated and transcribed again, that particular area or that coding for that particular protein in the next replication will jump into a new position. And so that's why we were calling them jumping genes. And then, there's this in the scientists, for example, jumping genes causes cancer, and that's a picture of Barbara McClintock. The DNA methylation, epigenetics, and I'm going to stop there. I want to tell you, because we will do question and answer, I do want to tell you about Benjamin. So he is a little boy with the congenital heart defect. We've still not identified why he did have a congenital heart defect. Because he had hypoplastic left heart syndrome, he required multiple surgeries. He's now about 4 or 5. And he has had 3 major cardiac surgeries. He's a delightful little boy. His mom went back to medical school, and so now she is, it's her time to do her residency. Benjamin's dad is an emergency medicine physician within our children's hospital. He has spoken both at in national meetings, and he's also gone to Congress to really try to engage the U.S. Congress in supporting more work regarding congenital heart defects. And so they are family medical advocates for the work that is being done. But the sad part is they still have, Benjamin is growing up to be able to do things, but his life will never be the same as a child without a congenital heart defect, and his life expectancy, of course, is much shorter. So I think that for the future that we need to continue to stay at a cutting edge of the genetics that we do, continue to try to identify new factors that cause birth defects, continue to really try to understand what the families want. It's one thing for me to sit in my lab and say, this is the next question that we should be asking. It's a little different to ask a parent, what are the most important research questions that you have? Since you have raised a child, or raising a child with a major structural malformation or a birth defect, what do you think we should do? What do you think the next questions are? And I think having more of a give and take between the patients, the parents, and the scientists is really important to be able to engage them. And so we hope to be able to do more of that as well. And the work that we did with the National Birth Defect Prevention Study is being continued in another new study that is funded by the CDC as well. And I'll stop there and ask if there are questions.
You mentioned several times structural defects. What about metabolic defects?
So there are a lot of metabolic defects that are very important that are not part of what I do, but that doesn't mean that they are very important, and our abilities to identify metabolic defects within children has increased remarkably with some of the new technologies in mass spectrometry. So, for example, in Arkansas, we used to just look for 4 inborn errors of metabolism. Now we're going to be looking, we moved about 4 or 5 years ago because of the technological advances to now being able to look at 29 inborn errors of metabolism. And we hope by this time next year we'll be looking at 32. So there's a real advancement there too. Yes?
Is anyone tracking, or do you ever get feedback on birth defects that don't show up until adulthood? They're minor. In my husband's family, for instance, they had a couple of cases of spina bifida. It was so mild it wasn't talked about, and nobody mentioned at the time. But when they went for military service, say, for a checkup, oh no, you have this, but never affected their life. Another case, it was a 20-year-old dying of a heart attack who had never been known to have, and I can't help but think that was, may or may not have been a defect, but is there anyone out there collecting information on that kind of thing?
Not in a systematic way, because we have to be able to identify them, but we certainly hear a lot of the stories of that, right? So that you'll have a young adult who will start to have some symptoms, maybe with exercise, and then we'll do ultrasound of the heart and an echocardiogram and see that in fact that individual had a defect that has been there all that individual's life, but does not become symptomatic until they're maybe in their 20s. And then they'll even have to have surgery. So, you know, it's not unusual to hear a story of somebody who has a small hole in the septum that are separating the two atrium, and that needs to be worked on. Just recently, I don't know why or how I came across it, but John Mellencamp, right? So John Mellencamp, the famous musician, right? He had a spina bifida and didn't realize he had it because he had surgery, but his parents didn't tell him to do it. He said, "Oh, don't worry about it." And he's now, 60, 62? And just within the last year or two, met the neurosurgeon who's 98, who performed the surgery on his back when he was an infant. So that's, I think that's sort of a notable story.
Is there a book available of talks that discusses what you're talking about this morning? Can you share with us?
There is a book called about malformations. It's a Smith book on malformations that I think is probably in its 5th or 6th edition right now. And then there are multiple new textbooks of the medical genomics, or the human genome is a nice one that gives more of a lay overlook, but a really well-cited reference for the chronological history called the human genome that I really like. So.
Do you see many defects that are passed down to the
Oh, that's such a good question. Yes, there are, and I think with the complex etiologies, but we will see more of that as well. That work for complex diseases is just in its infancy. There are certainly, in the pediatric population, we know of multiple neuromuscular disorders where it is the passage is the inheritance is from maternal mitochondria, and so that does happen. But in terms of looking at it in, say, for example, areas that we haven't looked at before, that work is just in its infancy.
I thought so this, it's like mother, or sister, but I can't get it.
Right.
Are there particular roadblocks that the field is coming across in advancing their research, like a particular black box or too much data, money, attitudes, or?
So one of the things is approach when you're looking for a rare disease and a rare variant. So there's a hypothesis that it's multiple different genes working together and each of them having a modest effect. And so when we start to look for modest effects, so if your effect size is small, your population or your sample size, in order to be able to test that hypothesis, needs to be very, very large. And so, and then the resources required to do a large population study is quite high. And so that is an issue. It is an issue to be able to, for example, with birth defects, just the obstacle of saying, if that tissue at that time when the heart is being developed, maybe the DNA methylation is different there when something goes wrong compared to if something is going right. But we can't get back to that. We can't get back to that critical time of being able to look at when something's going wrong. I think that's an issue. And I think that what you spoke about too is, at least when we take it to the clinical level, the huge amount of data
that we have. So if I have a baby that I've seen clinically, and then I ask for a microarray, which is different than a karyotype, right? I'm asking for, instead of just to look at the chromosomes, I'm asking for a look at the gene genetics, more at the individual gene level. I'll sometimes get a report back that will say, we see that there's a alteration, a variant, a mutation in this baby's gene on this chromosome, and the literature says that there's one study that has associated this with problems with development later on. Well, then what do you do as a clinician, right? Do you go in and you say to this mom, your child has this variant that there's one study, you can tell them that, and you would understand it if I went in and said that to you. But put yourself in a situation where you have, which is not unusual, you have a 16-year-old mother, you have her mom who maybe had her babies when she was 16, the level of, and hopefully not to be offensive or elitist, but maybe the level of education in the home is not high, and you say to them, well, your baby has a gene that's going to may cause this developmental disorder because we found it in one scientific publication. The nuances around that. So what do you do as a clinician? Do you give them that information, or do you just say, okay, nothing? And that's where I find it very difficult, because one would say, well, you're being paternalistic if you withhold the information. On the other hand, you're not being really, are you being wise if you give them the information? So that has to be,
They used to talk about the possibility of doing genetic counseling for couples that wanted to get married. And from what you've been showing me here, would that even be useful? There seem to be so many causes of birth defects that the old model that was in my head of two people that had, defects that matched up, get married, they have a baby, that baby has a birth defect, doesn't even seem to, does that even apply?
In Mendelian disorders, it does, right? In Mendelian disorders, where you still have autosomal recessive, autosomal dominant,
so how many
mode
of that,
of inheritance.
How much of that is this universe of birth defects you're looking at?
So within the complex etiology, complex etiology by definition is both that it's environment and genetics. But there are still babies that are born with specific, where there's a single gene disorder or a single chromosomal abnormality. The best one the one that I can think of off the top of my head right now is cystic fibrosis, right? So if I had a family one could say, well, if you wanted to have yourself tested and your to-be spouse tested, and you found out that you were both carriers of Delta 550 mutation, would that information help you to decide that maybe you wanted to adopt, or we are maybe that's an area that is really important. So the approaches that we take in terms of genetics are to look at candidate gene studies. So that is to identify a gene that might be involved in the metabolism of folic acid, for example. To look at pathways, so instead of just looking at one little gene, look at a comprehensive group of genes within a particular biological pathway. We now have whole genome association studies, which are also referred to as agnostic studies, where you don't come in with any a priori hypothesis about where the pathway is that is of importance, say, for the developing heart, but rather you say, okay, I'm agnostic, and you will see the term agnostic in the scientific literature. It's not just a term that I'm using it, it's really in the scientific literature saying, this is an agnostic approach, I don't have any beliefs about where this is going to happen, I'm going to put markers all the way across the whole genome, and then I'm going to let the data and my findings drive what I find.
I did 23andme.com along with dozens of other Mensa members. It was sort of a thing. And of course they cut that off, which just, I still don't understand why.
What did you find?
I found out I got darn good genes, but I'm just saying, that's the way they studied it. So Before the test came, I took at least 7 hours of online testing, of questionnaires. So they're gathering hundreds and hundreds of pieces of data from the online questionnaires, and then they're folding it into the other database of what your genetic material is. And I thought that's basically what they're doing.
Yes.
Yeah.
Yes, exactly.
You were studying for your genetic test?
Yeah, I got an A.
Probably cheated.
Well, I still got there.
We're now able to not just look at specific markers, or there's variations, but we're able to look, so we're not just looking now across the 21,000 genes and putting 5 million markers across those genes, but we're now able to look at the 3 billion individual base pairs, and that's called sequencing. And we can sequence just a particular area of real estate within the genome, or we can do it all the way across the genome, or we can limit ourselves to just doing coding areas, which are exons. So exons that code for particular proteins. The targeted deep resequencing is when we choose just a particular real estate area within the genome and look for just a sequence just